Dr. Kumar S. Venkitanarayanan is currently working as Associate Dean for Research and Graduate Studies, College of Agricultural, Health and Natural Resources, University of Connecticut. Dr. Venkitanarayanan worked as a veterinarian in India for a year before pursuing his MS in veterinary science. He also worked as an assistant professor at the Kerala Agriculture University, India for a year. Before taking up his current position at the University of Connecticut, Dr. Venkitanarayanan worked at the Center for Food Safety and Quality Enhancement, University of Georgia, Griffin, as a postdoctoral research associate for 2 years. His area of interest include understanding the molecular mechanism by which foodborne pathogens tolerate and survive adverse environment stresses encountered in nature, food, and in hosts. Activation of pathogenic microorganisms in foods using novel food-grade antimicrobials and studying their mechanism of action. Development of molecular methods for rapid detection of foodborne microorganisms. He bagged a lot of awards and recognition. He is nominated for membership in American Association for the Advancement of Science, CAHNR Kinsman Award for Excellence in Research, and received the Gamma Sigma Delta Junior Faculty Award. With these few words, I am welcoming Dr. Kumar S. Venkitanarayanan to this webinar. Thank you.
Thank you, Dr. Robinson. Good evening, everyone. It's a pleasure to join you all. Before I get started with my presentation, I would like to thank the Indian Veterinary Association, CVAS, and the Association of Meat Scientists and Technologists for organizing such a webinar in conjunction with the World Zoonosis Day. Can you guys hear me?
Yes, sir. Perfect.
Okay, thank you. And also I thank Dr. Sunil Beep for the personal invitation to be a participant in this webinar. So let me share my presentation now. Everyone see my presentation now?
Yes, sir.
Okay. So as you can see, the title of my presentation is Clostridium difficile, an emerging zoonotic pathogen. Clostridium difficile, it's not a new bacterium. It's been around for several years. In fact, the new name is Clostridium difficile, which is a new nomenclature added to that, but it is the same bacterium as Clostridium difficile. When I was a student in vet school in India, I don't remember learning anything about C. difficile as a part of our microbiology curriculum or even VPH curriculum. So I'm not sure if C. difficile is being taught to undergraduate veterinary medicine, the BVSC students in the curriculum. Anyway, Clostridium difficile is a gram-positive bacterium, actually. It is a very strict anaerobe. In fact, oxygen is very toxic to the bacterium. So it is very difficult to cultivate. That's why it got the name difficile. The Latin word means difficult. So it's extremely difficult to cultivate the organism in the lab. So that leads to under-detection of the bacterium in many countries. It is traditionally, it's a nosocomial pathogen, which means it's a hospital-borne pathogen. It predominantly, the infection have been reported in individuals who spend a long period of stay as inpatient in the hospitals, and also who receive a long-term antimicrobial therapy. However, that dynamics has changed, which I will talk about to you in the coming slides. Annually in the US, we get more than 500,000 cases of Clostridium difficile, which leads to an enormous medical cost, more than $6 billion as economic costs associated with medical costs or the healthcare costs. In light of its public health impact, the Centers for Disease Control in the US recently classified this bacterium, one among the 10 bacteria which produce significant human hazards. So it is currently classified as an urgent bacterium which requires significant scientific attention and control. In fact, the epidemiological data indicate that almost 1 in 9 patients who are above 65 years of age who get infected with Clostridium difficile results in mortality, actually. So see, as I told you, this bacterium, it's not a new bacterium. It's been around in the environment, in humans. In fact, it could be a part of the normal fecal flora of newborns. It has been detected in the gut. It has been shown to colonize up to 3% of healthy adult individuals without causing any disease. The key thing is only strains which can produce toxins, the toxigenic strains, can cause the infection in humans. So how it produces the infection in humans is when people undergo long-term antibiotic therapy, it potentially leads to a disruption of their normal gut flora or the gut microbiome. So when abnormal shifts happen in the normal gut microbiome, it leads to an opportunity for this bacterium to colonize. In fact, Clostridium difficile has been implicated in almost a quarter or 25% of cases of antibiotic-associated diarrhea in the US. Coming to the symptoms, the disease usually starts as a diarrhea, which progressively gets worse into a severe form of diarrhea. And then the critical symptom you see is in patients is something called pseudomembranous enterocolitis. Pseudomembranous colitis is commonly called. As you can see, it is characterized by severe inflammation of the colon with extreme thickening of the mucosa, which leads to loss of functionality of the colon. As you can see on the left panel, it's normal colon, versus on the right side, you can see the colon, which is characterized by pseudomembranous colitis. So this potentially leads to loss of functionality, cannot absorption of any nutrients, absorption of nutrients is impaired. So it leads to loss of functionality. And then there is another complication is there's a severe gas accumulation, which can lead to acute colonic distension or toxic megacolon, which can potentially lead to perforation of the colon. And finally, as you know, if the intestine breaks, ruptures, it leads to sepsis and finally death. So this graph shows the trend in the number of cases of C. difficile in the US during the 10-year period from 2000 to 2010. In fact, we are waiting for the next set of data from 2010 to 2020 sometime this year. So as you can see this in this graph, from 2000 to almost 2010, the number of C. difficile infections, especially in people above 50 years, have been steadily increasing during the 10-year period. And C. difficile infection is also reported in India, especially during the last probably 5 to 10 years. As you can see, there are many reports from India in the scientific literature. Clostridium difficile, a neglected but emerging pathogen in India. Prevalence of C. difficile infection among patients attending a tertiary care teaching hospital. Similarly, Clostridium difficile associated diarrhea in a tertiary care center. Another study on C. difficile associated diarrhea in children, especially associated with antibiotics. So it's been reported in India. And based on my interactions with researchers from India, and in fact, I was there in India last January to give a talk on C. difficile in the Indian Science Congress, which was held in Bangalore last January. Then I had an opportunity to meet with several people who work on C. difficile, and they're all, especially people from All India Institute of Medical Sciences. They told me that many a times C. difficile doesn't get detected. It is mostly underreported in India. So this, the whole transmission dynamics of C. difficile, once humans ingest the spores, C. difficile spores, because it's a spore former, the spores are highly tidy structures which can survive in the environment. So when the individual gets infected, the spores get, I will talk about how it results in the colonization. But once the person is infected, the person continuously sheds the spores of the virus in the feces, which results in the contamination of the environment, in contamination of fellow human beings, and it also results in the transmission into the community or in the hospitals, and also reinfection of the same person or the host. And here you can see the pathogenesis. So what happens is when a person undergoes prolonged antibiotic therapy, it is going to disrupt the normal flora. As you may know, the microbiome, the gut microbiome is increasingly recognized as very important for the whole health of a human individual. So when there is abnormal shifts happening in the different phyla of the normal intestinal microbial flora, that gives an opportunity for many pathogens, especially C. difficile spores, when they are getting ingested, results in the germination of the spores. And to add germination more possible, what happens is the bile acids which are secreted in the intestine, they act as a stimulant to stimulate the germination of the Clostridium difficile spores in the intestine. So the spores germinate, vegetative cells of C. difficile are formed, and which colonize the large intestine, and they produce two potent toxins called the toxin A and toxin B, which leads to severe inflammation, diarrhea, pseudomembranous colitis, and other symptoms. So how if you look into the genome, there is a critical area in the genome of C. difficile, which is called the pathogenicity locus. As you can see in this diagram, it has two critical genes, TCD-A and TCD-B. These two genes secrete the potent toxin A and toxin B, which are the primary virulence factors in this bacterium. Then there is another gene called the TCD-E, which secretes a protein which is responsible for secreting the toxins out of the bacterial cell. And then there are two controllers which control the synthesis of the toxin. The one is TCD-R, which is a positive regulator of the toxin synthesis. And there is a TCD-C, which is a negative regulator of the toxin synthesis. So basically, when these genes harmoniously work, the toxin production is increased, toxin production is cut off. So that's how the whole toxin synthesis is controlled. So what is the treatment strategy against CDA, or say Clostridium difficile infection? In spite of the fact that antibiotic therapy leads to the occurrence of Clostridium difficile infection, antibiotics are still the drugs of choice for treating CDI. In fact, there used to be a number of antibiotics, including clindamycin, metronidazole, vancomycin, fidaxomicin. They were all used for treating Clostridium difficile, but metronidazole and clindamycin are no longer commonly used because most of the C. difficile are resistant to these antibiotics. Currently, the two commonly used antibiotics include vancomycin and the new antibiotic called fidaxomicin. In fact, there are reports now in the literature that vancomycin-resistant C. difficile have emerged. So antibiotic resistance emerging in this C. difficile is another major concern among physicians, actually. So in addition to antibiotics, especially currently the two limited antibiotics we have, probiotics, as I told you, the disease onset is due to the disturbance in the normal gastric flora, actually. So probiotics, not a single probiotic species, a mixture of probiotic species are administered to patients to restore the normal flora. Sometimes the probiotics, it's been more than 50% of the cases, the probiotics may not be fully effective. So the next strategy employed is fecal transplantation. This is very commonly done in the US hospitals, where they transplant feces from normal human beings to the patients so that it can transfer the whole set of normal microflora to resist the C. diff, Clostridium difficile. And in extreme cases, colectomy, the cutting and removal of the affected part of the colon is practiced. So currently in the literature, if you see, the ideal strategy, because antibiotics are not going to be working fully effective because it can contribute to the disruption of the flora and because of the antibiotic resistance. An ideal strategy that has been increasingly explored in the literature in the medical field is something, a drug which is used for C. difficile should not disrupt the normal flora, because that's what the starting point of the infection. Then it should reduce the toxin production by the bacterium, because that is the most important culprit which produces the infection. Then it also should inhibit the Clostridium difficile sporulation and spore production, because spores are critical for the transmission and reinfection of the same patient. And unlike antibiotics, any new drug you use against Clostridium difficile should not induce resistance. There's a lot of interest in coming up, identifying new drugs against this bacterium, including my own lab. I have two or three PhD students who have worked on this bacterium, including a former student from CVAS, Dr. Kumar S, who is currently a professor in Iowa State University School of Veterinary Medicine. His whole dissertation was on Clostridium difficile, and he identified two major phytochemicals which did not disrupt the normal flora and decrease the two virulence factors, namely toxin production and sporulation. So that is a new strategy which is currently explored, including my own lab. So now let me switch gear and talk about some new trends which are observed in the C. difficile epidemiology worldwide. And excuse me, can you see the slides?
No, sir. Something over. Now it's okay.
Can you see the slides?
Yes, yes.
Okay.
Yes.
So in terms of some of the new trends which has happened in the C. difficile epidemiology worldwide is a hypervirulent strain of Clostridium difficile, not strains of Clostridium difficile have been reported in the US and in many other countries. The one in the US is called NAP1, which is North American pulsotype 1 or the ribotype 027. What is the characteristic features of this hypervirulent strain is it produces large amount of toxins with a very high degree of sporulation. And it is also resistant to many clinically used antibiotics. So these are all disturbing trends in the epidemiology, because the treatment strategies against the hypervirulence Clostridium difficile, it's very rigorous treatment we have to adopt, and it's not very easy. Another disturbing trend which has been increasingly observed during the past several years in C. difficile epidemiology is that, traditionally I told it's a nosocomial pathogen associated with hospital-borne infection. But that has changed now. Now, more and more cases of Clostridium difficile are community associated. That means they occur outside the hospital environment. In fact, more than 1/3, almost 30 to 50% of the current cases of Clostridium difficile are in the environment. They happen in the community and outside the hospital environment. And it is interesting to see that these cases occur in the community in people with no recent hospitalization history and no history of antibiotic therapy. So there should be something which is propagating the transmission of Clostridium difficile in these community patients. So that's triggered interest in many researchers to looking into the driving factors which can propel the community-associated C. difficile infections. So some of the factors which have been proposed include there could be spread between the hospitals and the community by people, by traffic of people who are not only patients, but also bystanders or people who bring patients, etc. So that could be one potential chance. And there could be asymptomatic colonized hosts within the community. within the community. So they don't show any symptoms. So they don't get recognized and treated. So they could be spreading the disease within the community. And there could be other non-hospital healthcare places like elderly homes, facilities, centers where taking care of elderly patients, nursing homes, etc. There, if infection is there, it could spread to the community much rapid rate. Then there could be household transmission is very high. If one patient within the house gets infected because of the fecal oral route transmission due to the spores, it could be high. Then there are many environmental reservoirs of Clostridium difficile. C. difficile has been detected in the soil, in water, in lakes, etc. So they could be potentially contributing to the community-associated infection of Clostridium difficile. And another major factor we all know in community infections, foodborne infection could be a potential source. And the last one could be, there could be some zoonotic agents, or there could be some source of zoonotic potential within the community. So let's look into C. difficile in animals. Clostridium difficile has been detected from pretty much all domestic animals and birds, actually. In fact, many toxigenic human isolates of cattle of C. difficile have been isolated from cattle, horses, pigs, and pets. As we know that Clostridium difficile produces neonatal diarrhea in piglets and in calves. There was an interesting thought from scientists. Can animal-derived foods could be a potential source of C. difficile? So there was a lot of interest in this, and worldwide, people have been looking into the prevalence of Clostridium difficile spores in animal-derived foods. So I'm summarizing, because of the lack of time, I'm summarizing the data on the prevalence of Clostridium difficile, which is out in the literature. Are you able to see until this slide C. difficile in animals?
Yeah.
Okay, then as I told you, there was an interesting thought from the scientists, since many farm animals harbored Clostridium difficile and neonates, especially piglets and calves, could be infected with C. difficile. Could animal-derived foods could be a potential source of C. difficile. So that triggered prevalence studies throughout the world, and I'm just summarizing the data from the prevalence studies in the literature, which showed that Clostridium difficile prevalence ranged from 0 to 42% in one study. Similarly, 2 to 41% in chicken and seafood. So once we looked into the prevalence studies, and in fact, as a part of, I told you, Dr. Shan Kumar, who was a student, we also did prevalence studies in the US, and we found very, very low prevalence in pork and beef, and also in vegetables, actually. So there is no direct evidence showing that foods could be a potential source of C. difficile to human. There have been no foodborne outbreaks of Clostridium difficile. In fact, there have been no case control studies where you can see that a particular food which had a Clostridium difficile, the same Clostridium difficile, phenotypically and genotypically, exactly the same C. difficile could be detected in a patient. So with the lack of direct evidence pointing towards the presence of C. difficile and a potential role of food as a source of C. difficile, scientists started looking into the zoonotic potential, which is could C. difficile be in pet animals. We all know that there is a close companionship between pet animals and humans, and especially, we tend to, pets, especially in Western countries, because of the weather, they are kept inside the home all around the year, actually. So there's a very close interaction and association, and pets are as household members, and there is a great potential for the fecal to oral route. For example, humans, there's a human to human transmission happens fecal to oral route because the spores are excreted in the feces. The same scenario could be applied to pets. If pets get infected, they could discharge the spores in the feces. So that could result in infecting humans within the household. And the other observation is critical to emphasize here is similar antibiotics have been used in small animals and humans for treatment generally. So this could, the similar antibiotics, they give a selective advantage resulting in the selection of C. difficile strains, which are host-adapted for both pets as well as humans. So thereby, the same C. difficile isolates or strains could be interchanged between the humans and the pets, or pets and the humans. So in fact, in the US, there's a strong argument put for screening household pets for C. difficile as a public health factor before you resorting to treatments of humans, actually. So as I told you, this triggered a lot of interest from worldwide, actually, but there have been very, very few studies looking into the prevalence of C. difficile in pets. As again, I have summarized the data from the literature. There have been around 2 or 3 studies been done worldwide looking into the prevalence of C. difficile in dogs. Most of them were very small studies. The data, including done by Dr. Kumar and my lab, the data range from 4% to 30% of C. difficile presence in dogs, actually. And in the literature, you can see that C. difficile toxin A and B, or both, can be detected in approximately 20% of the dogs, either outpatient or inpatient healthy dogs, as well as dogs which show diarrhea. And there was another study which showed that almost 90% of the puppies, they show C. difficile in their feces at least once during their first 10 weeks of their life. And almost 50% of the isolates from the pets, especially dogs, are toxigenic, resulting in potential for them to produce a severe infection in humans. And then as a follow-up to that, people started looking into, because cats are also kept indoors, as you know, and cats have a much higher, greater chance of spreading the disease because the cat litter box is usually kept within the house, actually. So what happens is the cat defecates and it is in the litter box and it is changed once a week, actually, or a few days, within a few days once. And the spores could be spreading within the environment, within the house, and it could be transmitted to humans. But however, there have been very minimal studies on cats, and there needs to be more information is needed from cats. But based on the current literature, in 1 or 2 papers, the carriage rate of C. difficile could be the same in, or it could be different, because in different studies we have seen different results. So we did, as I told you, in Connecticut, we just started a study, prevalence of C. difficile in dogs and cats in Connecticut. We just started a couple of months ago. In fact, the person who is working is Dr. Abraham Pellissieri, who is again a graduate of CVAS, so he's a postdoctoral scientist in my work, in my lab now. In fact, his PhD work was on C. difficile. So again, I just wanted to let you know that it is a very recently started. We don't have a lot of samples, but I will show you how we detect the C. difficile in the samples. As I told you, it is not an easy task to detect this bacterium. So I will show you the methods which we use. So we use the collector samples, the feces samples, and we inoculate it in brain heart infusion broth. Then we incubate it 7 days in an anaerobic chamber. This, as I told you, even the slightest concentration of oxygen is toxic to the bacterium. So we were fortunate to have a very fancy machine which highly computer controlled, which controls the whole gaseous environment the pressure, the humidity, everything is controlled very precisely. So this helps to detect the bacterium. So we incubate it in the anaerobic chamber for 7 days. Then we take the tubes for after 7 days and keep it at room temperature to stimulate the sporulation. Then, excuse me, we treat the culture with ethanol. Ethanol is done to kill all the other bacteria and vegetative cells of the bacteria. And then we centrifuge them to separate out the spores, the Clostridium difficile spores. Then we inoculate them in a very specific medium, cycloserine sulfoxafine fructose agar, which contains sodium taurocholate. As I told you, bile salt is a stimulant for sporulation. So we add that. Once we have the colonies, we test it for an enzyme called proline aminopeptidase test, because proline aminopeptidase is an enzyme produced specifically by many Clostridium difficile, not by most of the other C. difficile, Clostridia. And following that, we get the pure culture, and then we isolate the DNA and do a real-time quantitative PCR for the toxins to make sure that it has a toxin A and toxin B. Then we collect the DNA and do the ribotyping, and we also do a cytotoxicity assay of the culture supernatant, because if it is C. difficile, it produces toxin A and B. The culture supernatant should be cytotoxic to vero cells. It kills the cells, cytopathic effects produced. And then we take the culture supernatant and do an ELISA also to detect the proteins or detect the toxins. Following that, we do capillary electrophoresis to confirm the ribotype, and we do WGS or whole genome sequencing to make sure it is C. difficile. So this is, in a nutshell, the steps which are used to confirm Clostridium difficile. So based on this, our preliminary data, total samples of in Connecticut, we have analyzed 54 dog samples from veterinarians around the state, because both animals which are brought to the pets, healthy animals, and also animals, if they have any disease condition, we screen the feces. So right now we have analyzed 54 dog feces samples and 39 cat samples. We have found 14 canine samples positive for C. difficile by qPCR, of which 9 are toxigenic, both produce toxin A and B. 3 cat samples have been positive, of the 2 are considered toxigenic. So the preliminary inferences are approximately 17% of dog, cat, and 55% of C. difficile from cats have been C. difficile positive and also toxigenic. And the initial molecular characterization suggests that probably there's a zoonotic risk, because we have not annotated the genome. Based on the genome, there could be this identical regions, so suggesting it could be the potential strain. And also in cats, the number of samples we have analyzed is too small. We are planning to go up to about 200 to 500 samples for both dogs and cats. There's a high sample size is needed. And again, we have to annotate the whole genome sequence data to see if there are identical regions, identical sequences between human isolates and the pet isolates, to see if there is a jumping of isolates from the dog to the humans. So with that, what I would like to conclude is, Clostridium difficile infection is in fact a significant public health concern, actually. And incidence rate and disease severity are constantly increasing in many countries worldwide. And recent reports from many parts of the world indicate that pets could be a potential zoonotic source of this bacterium. And also detection of C. difficile, which are similar both in humans and pets, underscore or emphasize the potential zoonotic potential of this bacterium. So currently, as I told you, we are planning to do more studies. And what we are planning to do in the future is, we are planning to collaborate with the hospitals in Connecticut and also with the health department to see if any patient who comes, human patient comes for C. difficile, if we could interview that patient and see if there are pets in his or her home, and we could get a sample from their pets and see if they have the same C. difficile. That would be a clear, and once you sequence the genome, you would get a clear data that that C. difficile came from the pet, if in fact there is a transmission. And also, there are reports that the toxin genes and antibiotic genes in C. difficile are mobile. That means they can be transferred from one species to another by horizontal gene transfer. So many genes from human or pet C. difficile could be acquired to human C. difficile, and that could complicate the situation in terms of antibiotic resistance. And as I told you, there is more concrete data needed to confirm that especially cats are indeed a potential zoonotic potential of C. difficile. With that, I would like to limit my talk within that time. And also, since I have a meeting right after this, I would like to conclude my presentation. I'll be happy to answer any questions you have. Thank you for your attention.
Thank you very much, Kumar. Very interesting topic. You have educated us. It's a new topic. We have learned a lot of things from you. Thank you very much.
So may I ask, is it Dr. Robinson or who is?
Yes, I'm Dr. Robinson.
Dr. Robinson, hope you remember me.
Yeah, I have seen you.
Yeah, I would like to ask all of you, is still Clostridium difficile, how much of Clostridium difficile is taught in the veterinary curriculum nowadays?
I don't think so.
No, I think Clostridium difficile finds its mention when you are teaching Clostridium.
That's it?
That's it. That's the way it is going. But actually, it's nice to hear from you that at least it has not got much of a problem with our food safety angle, because we are finding more and more pathogens here.
Exactly. Although we have been, there have studies which have detected C. difficile on foods, there is no direct evidence, not even a single outbreak during the last 20 years. And they couldn't match with a case control study. So although people would say there is a potential, there is no concrete data supporting that. And the number of cases based on, as I told you, when I attended the Science Congress to give the talk on C. difficile in January, in my interaction with some of the scientists from all lines of medical sciences, especially who are infectious disease specialists, they told me that the number of cases of C. difficile have been increasingly happening, but many a times it doesn't get reported because it is not detected finally. But some of the isolates which are sent to the lab for in-depth characterization reveal that they are Clostridium difficile. Usually they are categorized as unknown C. difficile.
What exactly could be the source of such C. difficile, which again it is present in the human beings?
I mean, again, it could be right now the question we have is, with long-term antibiotic therapy, because C. difficile could be a normal part of human flora, healthy part of normal flora. There have been people who are known to carry this. So when the antibiotic therapy happens, or somehow the gut flora imbalance happens, they could happen. Or they could be ingesting it from an environment source or from pets, who knows. So there is no clear-cut data now showing how the community transmission is happening. And almost 40% of the outbreaks of C. difficile in the US are now community oriented. And in the US, I hope the same in India to some extent, dogs and cats are completely indoor. because of the weather and so many factors.
Now, another question which crops up is, because of the prevalence rate which you have been mentioning and the humans as normal carriers of this organism, would it not be prudent to, screen the human population? How much percentage they are carrying this in as a normal microflora?
Yeah, so because that data is there. There are many papers which have shown that around 3 to 5% of normal adults carry this. That's it.
Okay.
And it is more often seen in infants, and as they grow out and their microflora develops, C. difficile disappears.
Okay. So I think it's a fascinating organism which is creating some problems.
Yeah, there's a lot of funding potential for this organism actually now in the US.
Right.
Any other questions?
Very interesting topic.
Thank you.
Dr. Arshad?
I would like to call our co-chair, Dr. Vrinda Menon, for sum upping this particular session.
Thank you, sir, for Clostridium. Not in our syllabus to believe it to be not very zoonotic, but after hearing your talk that we need to go in for work related to zoonotic prevalence in care.
Yeah, actually,
thank you very much.
Yeah.
Yes.
Sir, there is some questions in the chat box. I just read it out.
Sure.
From Justine Kaur. Sir, please mention the enzyme and its role.
Pardon? I didn't hear you.
The question is, sir, please mention the enzyme and its role.
Yeah, because C. difficile, most of the C. difficile isolates produce an enzyme called aminopeptidase, proline aminopeptidase. This enzyme has been reported chiefly in C. difficile, and so it has been used as there is a disc which is used in the diagnosis of C. difficile. What you can buy is you can get a small disc impregnated with the substrate proline, and what you do is get a supernet into the cultures and add, if that enzyme is there, it will produce a color change because it breaks the substrate, because the proline aminopeptidase is believed to be produced only by C. difficile, but in our experience, we have found that at least 2 other Clostridia produce the same enzyme. So this proline aminopeptidase is a specific enzyme test used for the detection of C. difficile.
And there is a question from Dr. Parminder Singh. Is it a self-limiting disease in children?
In fact, it is not a self-limiting disease because in children, what happens is, especially in neonates, this opens, a lot of discussion topics. The role of breast milk feeding in contributing to the development of a microbiome in infants, and that provides resistance to pathogens, including C. difficile. There have been reports based on that. So it is not a self-limiting disease in humans unless you have a well-developed microbiome or well-developed gut microflora.
And there is a question from Masianna HM. Sir, are there any case report from wildlife?
Any case reported? Yes, wild animals have been. There are, I don't remember the top of my head, there are reports of C. difficile from wild animals. Yes.
Hello, Dr. Varsha Unni, are you able to ask directly? Please unmute and ask.
Hello.
Hello.
Sir, I'm audible?
Yes,
Yeah.
please.
Yeah. Thank you for such a wonderful lecture. So
You're
my
welcome.
doubt is, as you said, the treatment with antibiotics is a risk factor for the disease. So what all alternative approaches can be adopted for tackling this infection, and whether any novel immunotherapeutic agents are available, sir?
To my knowledge, as of now, there is no effective immunotherapeutic agent. But as I told you, the probiotics are a major option. That too, not one probiotic, collection of so many different types of there are commercial preparations available, and fecal transplantation that gives the whole microflora from a healthy individual. And there are many other drugs. For example, my lab has we have a couple of drugs, plant molecules, which have been shown to reduce the symptoms, which have been shown to reduce the toxin production, sporulation, without adversely affecting the microbiome or microflora. So there is a new strategy called anti-virulence strategy, which is basically targeting to control C. difficile, not to kill it or not to reduce its growth, because only when a bacterium is challenged with a drug to kill it or to reduce its growth, the bacteria have the ability to come up with a resistance mechanism. When their growth or their existence is not challenged, they do not come up with resistance mechanism. So the anti-virulence strategy is basically to identify molecules which will target the virulence factors, like reduce the toxin, reduce the sporulation without affecting its growth or without affecting killing it. So that is a new strategy which is being evolved. So that's what I showed in my slide. Anything which does not affect the growth, especially the C. difficile or even the normal microflora, obviously it doesn't affect. If it reduces the toxin, it reduces the sporulation, it would be the most ideal drug against C. difficile. So that a lot of people are interested in identifying the molecules, including my own lab. We have screened so many different molecules, including probiotics, including supernatants from probiotics, molecules produced by probiotics, phytochemicals. We have been successful in identifying 2 phytochemicals in that, and we have completed with the mouse study. Now we are planning to move on with large animal swine as a model before collaborating with a medical school to see if there could be any potential human clinical trials, because the 2 phytochemicals which we are working against this are food grade molecules.
Thank you very much.
You're welcome.
I think there is no more questions, sir. Thank you so much, sir, for a very informative session. And now I invite Dr. Sunil Kumar S for a lot of time.
Thank you, Dr. Risha. So, completed the 15th session of the international webinar, and we are running short of time anyway. So, on behalf of the organizers, the Indian Veterinary Association, Meat Technology Unit, Kerala Veterinary and Animal Sciences University, and the Association of Meat Science and Meat Scientists and Technologists, India. We thank Dr. Kumar S, Venkitanarayanan, sir, the Associate Dean of Research and Graduate Studies, Professor, College of Agriculture, Health and Natural Resources, University of Connecticut, for delivering such an excellent talk on is Clostridium difficile in an emerging zoonotic pathogen. Thank you so much, sir, on behalf of the organizing committee. Then I thank Robinson J.J. Abraham, sir, who chaired the session. On behalf of the organizing committee, we thank you, sir. Then I thank Madam for co-chairing the session on behalf of the organizing committee. Thank you. Thank you so much, Madam.